Hello, my name is Ryan Guyverlick, and this is my final project on the cell membrane for Bio 162. The cell membrane is composed of many different molecules, but the three most important molecules are phospholipids, proteins, and cholesterol. Phospholipids, shown here, contain a hydrophobic hydrocarbon tail and a hydrophilic phosphate head. Because it is both hydrophilic and hydrophobic, it is considered to be amphipathic. Proteins are embedded across the cell membrane. Integral proteins go completely from one side of the membrane to the other side. Peripheral proteins are only embedded on one side of the membrane, shown here. The cholesterol in the membrane helps restrict the passage of molecules by increasing packing of phospholipids. It takes up space and fits into spaces, and this prevents hydrophilic material from diffusing across the membrane. One of the most important roles of the cell membrane is selective permeability. As mentioned before, phospholipids are both hydrophilic and hydrophobic. When a double layer is formed, such as right here, this creates a membrane that has hydrophilic ends facing outside and hydrophobic ends facing inside. Because the inside is hydrophobic, small hydrophobic nonpolar molecules can diffuse across the membrane. One example would be estrogen, shown here. This is a nonpolar molecule, so it is able to diffuse across. Gases are also able to diffuse across freely, such as when oxygen is released from the hemoglobin and goes into the cell. However, due to the hydrophobic center, hydrophilic charged molecules cannot diffuse across. This includes ions such as potassium, sodium, and calcium ions. This way, the cell can prevent unwanted material from entering the cell. Another role of the cell membrane is communication. Cells communicate with each other through the molecules and chemicals. For instance, neurons communicate with each other by releasing neurotransmitters over a synapse. Synapses are very tiny spaces between neurons, allowing for quick communication. Glands communicate with cells by releasing hormones. Hormones are released into the bloodstream and take a little longer to reach the target cell. Both of these communication methods are monitored by surface proteins. One example is shown here. These proteins remain on the outside of the cell and bind to molecules to induce a change within the cell. So how do these concepts apply to what we have learned in lab? Neurons transmit electrical impulses to induce an action potential and release neurotransmitters over a synapse. These neurotransmitters can be acetylcholine, which bind to the receptors on the outside of muscle cells to cause contraction. Ions are required to create action potential, but they are charged, meaning they cannot cross the cell membrane. The neurons require integral transport proteins for ions to cross from one side of the membrane to the other. Without these proteins, ions could not cross the membrane, and an action potential would never be created. These proteins are ion-specific, so the cell can control which ions enter the cell and which ions do not cross. In this diagram I drew, this shows how the proteins are required for charged particles to cross the membrane. Because of the hydrophobic center, ions cannot freely diffuse across the membrane. In this diagram, red circles are potassium ions and green spots are sodium ions. The ions can only cross the membrane through their specific ion transporter protein. Thank you.